It's really a honor to be here today to present some of our results about the biology of the mesenchymal stem or stromal cell in deficient animal for P4i. We are working for many years in this molecule and in this cell. F and Ephrin, the ligand, are involved in many processes controlling the development and also governing the tissue homeostasis by regulating the attachment, detachment of the cell. And mesenchymal stem cells are currently the most used cell in cell therapy. So we analyze by quantitative RT-PCR the expression of different transcriptional factors directly related with the osteogenesis, with the production of bone cell. And we found that in the animals that are deficient for this P4i, the amount of this transcriptional factor, but also the production of the bone demonstrate by staining with alizarin red increase very, very importantly. More relevant was to demonstrate that in these knockout mice that obviously cannot transmit signal through this receptor, the mice were unable to develop an osteoporotic phenotype. So we induce osteoporosis in wild-type animal just by ovariectomy or after treatment of glucocorticoid, concretely dexamethasone, and we analyze comparatively this condition in wild-type animal and in the animal deficient in P4i. You can see here the difference in the wild-type condition that obviously they develop an osteoporotic phenotype, and by the contrary, our mutant have no anatomical manifestation of this feature. Also, we evaluate the presence in the serum of CTX1 and in the urine of the calcium that are parameters that reflect the release of this molecule in the blood. And so you can see that the animal, the mutant animal have no decrease in the amount of this parameter. We analyze also what kind of cell could be involved in this process. And you know that the osteoporosis is a condition resulting from the balance between the activity of the osteoblast and the osteoclast. Well, in this animal, we demonstrate that the proportion of osteoblasts are increased in the mutant mice. And by the contrary, there is a significant decrease in the number, in the proportion of osteoclasts. So the result is the no appearing and developing of the osteoporosis. So we try to translate this result to the human condition, and in vitro we demonstrate, obtain exactly the same result that in our mutant mice. In this condition, we isolated human mesenchymal stem cell from the bone marrow or from the adipose tissue, and block the FB3 signaling using different molecules that include fusion protein or antiserum or monoclonal antibodies specific to B3. You can see again that in several of these conditions, the expression of the marker for the bone cell increase in this experimental condition. Not only different transcriptional factor, but also the expression of alkaline phosphatase or the deposit of calcium that you can see here and is quantified here. So our proposal is that really we can manipulate through the signaling of FB and Ephrin B human mesenchymal stem cell, and to use for faster healing of the bone fracture or to improve the condition of osteoporosis. We have some preliminary results in mice that support this hypothesis. Thank you for your attention.